Hello everyone, welcome to e-learning platform of agriculture and GK. My name is Jigyasha and today we will be discussing about topic food microbiology, which is a part of syllabus for FSSAI exam and state food safety officer exam. So now before moving with the video, I want to tell you about these courses curated by agriculture and GK. So if you are preparing for FSSAI assistant or FSSAI central food safety officer or technical officer, you can enroll for these complete courses where you will get 6 to 7 ebooks which will cover your entire syllabus, YouTube class notes, 1 question bank, 10 full-length mock tests with All India ranking, CBT 1 and 2 full notes, discussion group and personal guidance. And the price for the courses for FSSAI assistant it is 1500 and for Central Food Safety Officer or Technical Officer it is 1700, but you can get a discount of 25% by using this coupon code. You can enroll for these courses through the link given in description box or you can download that from Play Store as well. So now moving with the video, here is our first question. Spoilage in food because of microbial activity can be prevented or delayed by prohibiting the entry of microorganisms in food, physical removal of microorganisms, hindering the activity of microorganisms, all of the above. So जो फूड में माइक्रोबियल एक्टिविटी की वजह से जो स्पॉइलेज होती है उसको प्रिवेंट और डिले करने के लिए इफ़ वी आर prohibiting the entry of microorganisms in food, it can be the spoilage can be prevented. And if we physically remove the microorganisms, then also, and so can be done with hindering the activity of microorganisms. So our answer will be all of the above. Option D is the right answer. Now moving with the next question. The growth of aerobic food spoilage and pathogenic microorganisms can be suppressed by humectants, exhausting, both A and B, none of the above. So the aerobic food spoilage is when the food gets spoiled in the presence of oxygen. So how can we prevent it? So we know the pathogens will grow in a high moisture area or in an area where there is a ample amount of oxygen, since we are discussing about aerobic food spoilage, so we will need oxygen. So by using humectants we will reduce the moisture content, so it can be used to suppress the pathogenic microorganisms and prevent aerobic food spoilage. And in exhausting we will remove the air. So if we won't let the air enter the food, so there won't be an aerobic atmosphere for the pathogens to grow. So our answer will be humectants and exhausting, both A and B. The growth of aerobic food spoilage and pathogenic microorganisms can be suppressed by humectants and exhausting. So the right option will be option C. Now the next question is, which of these bacterial components is least likely to contain useful antigens? Cell wall, flagella, ribosomes, capsule. So इनमें से कौन से जो bacterial component है, it will contain less useful antigen. And we know cell wall, flagella and ribosome, these three are very important when it comes to antigens. So these can't be our answer. Our answer will be capsule. Capsule is least likely to contain antigens. So our answer will be option D. And antigens कौन से होते हैं? These are the disease causing substance or organisms that enter the body, and when the antigens enter the body, our body starts making antibodies. So capsules are least likely to contain these antigens. And how these are useful? Because the body will make antibodies to fight a certain disease. So antigens help in creating the immunity, innate immunity. So our answer is here D. Capsule is least likely to contain useful antigens. Now moving next, which of the following contains structures composed of N-acetyl muramic acid and N-acetyl glucosamine? Mycoplasmas, amoeba, E. coli, spheroplast. So which one of these contains these structures? Contains a structure that is composed of N-acetyl muramic acid and N-acetyl glucosamine. So it is E. coli or Escherichia coli. So our answer will be option C, E. coli. E. coli composed of N-acetyl muramic acid. No, E. coli contains the structures that are composed of N-acetyl muramic acid and N-acetyl glucosamine. So our answer will be option C. Our next question is, the association of endotoxin in gram-negative bacteria is due to the presence of steroids, peptidoglycan, lipopolysaccharides और polypeptide. So जो biological activity है endotoxin की, it is associated with lipopolysaccharide, and the toxicity which is associated with lipid component, and immunogenicity is associated with the polysaccharide component. So here our answer will be lipidopolysaccharides, because the immunogenicity जो है that is associated with polysaccharide component, और जो toxicity है that is associated with lipid component. And this association with gram-negative bacteria is associated with the biological activity of these endotoxins. So our answer is option C. Now moving next, the prokaryotic cell membrane contains metabolic enzymes, is selectively permeable, regulates the entry and exit of materials, contains protein and phospholipids. So जो हमारे प्रोकैरियोट्स होते हैं वो होते हैं सिंगल सेल, जो हमारे होते हैं वो प्रोकैरियोटिक, जिन्हें हम प्रीमिटिव सेल्स भी कहते हैं, जो ओल्डर सेल्स जो है हमारी. सो जो प्रोकैरियोटिक सेल मेम्ब्रेन है, it contains proteins and phospholipids. Metabolic enzymes, selective permeability, and regulation of entry and exit of materials is in eukaryotic cell membrane. So our answer will be option D. It contains protein and phospholipids. Now moving next. Our next question is, which of the following statements regarding gram staining is wrong? Mycobacterium tuberculosis stains blue because of the thick lipid layer. Streptococcus pyogenes stains blue because of a thick peptidoglycan layer. Escherichia coli stains pink because of thin peptidoglycan layer. Or Mycoplasma pneumoniae is not visible in the gram stain because it has no cell wall. So we know the Mycobacterium tuberculosis has a waxy coating its cell surface because of presence of mycolic acid, and this coating makes the cell impervious to gram staining. So this statement is wrong. Streptococcus pyogenes stains blue because of thick peptidoglycan layer. This is true. E. coli stains pink because of thin peptidoglycan layer. This is also true. Mycoplasma pneumoniae is not visible in gram stains because it has no cell wall. That is also true. So our wrong statement is option A. Now moving next. Which of the following is not a recognized cause of diarrhea? Vibrio chlori, E. coli, Clostridium perfringens, Enterococcus faecalis. So which one of these is not a reason for diarrhea? So no. So we know Vibrio chlorei, E. coli, and Clostridium perfringens, they causes diarrhea. So Enterococcus facilis doesn't cause diarrhea. So our answer is option D. Now moving next. Our next question is, which of the following is a gram-positive U bacterium? So we have already discussed कि gram positive bacteria कौन से होते हैं, gram negative कौन से. So if you haven't watched the previous videos, you can go and check them. So now our options are Actinomyces, Clostridium, Rhizobium, Clostridium and Actinomyces. So here Clostridium and Actinomyces, it also gives gram positive test, and it also Clostridium also gives positive result in the gram stain test. So our answer will be Clostridium and Actinomyces. So option D is the correct answer. Now moving next, which of the following microorganisms is not responsible for urinary tract infection? We commonly say UTI. So which one of these microorganisms is not responsible for UTI? Proteus mirabilis, E. coli, Klebsiella pneumonia, they causes UTI. E. coli is a common disease causing organisms. It causes diarrhea as well, UTI also, nausea and other disease also. So what's left? Bacteroides fragilis. This doesn't play a role in causing UTI. So our answer will be option D. Bacteroides fragilis. Now moving next. Which of the following is diagnosed by serologic means? Actinomycosis, Q fever, pulmonary tuberculosis, gonorrhea. So serologic means, it means testing the blood serum. जो blood serum हमारा जो blood होता है, testing the presence of a disease causing bacteria in our blood using the serum. So that is serologic means. So जो टेस्ट होते हैं जिसमें ब्लड सैंपल लिया जाता है, these are serologic tests. So which of these diseases is diagnosed by serologic means? So it is Q fever. Q fever is diagnosed by serologic means, and Q fever is caused by Coxiella burnetii. This is also a very important question. Q fever is caused by Coxiella burnetii. So our answer is option B. Now next question is, diarrhea is not caused by Shigella dysenteria, Streptococcus pyogenes, Clostridium difficile, Salmonella enterocolits. So हमें पता है these three are responsible for diarrhea. And जो Streptococcus pyogenes है, it causes scarlet fever and pneumonia. जो pneumonia होता है हमारा, that is caused by Streptococcus pyogenes. So our answer will be option B. Now moving next, our next question is, the coagulase is done to differentiate Staphylococcus aureus from Staphylococcus epidermidis, Staphylococcus epidermidis from Neisseria meningitidis, Streptococcus pyogenes from Enterococcus faecalis, Streptococcus pyogenes from Staphylococcus aureus. So the coagulase test is done to differentiate Staphylococcus aureus, that is gram positive, from coagulase negative staphylococci or Staphylococcus epidermidis. So coagulase is an enzyme, and ये एंजाइम प्रोड्यूस कौन करता है? इट इज प्रोड्यूस्ड बाय Staphylococcus aureus that converts soluble fibrinogen in plasma to insoluble fibrin. So जो coagulase test होता है, it is done to detect free coagulase. And here our answer is option A. It differentiates Staphylococcus aureus from Staphylococcus epidermidis. That is positive and that is negative. Gram stain positive, gram stain negative. Now moving next, prokaryotic cells are more resistant to osmotic shock than eukaryotic cells because their cell wall is composed of peptidoglycan, they are selectively permeable, they contain osmoregulating porins, they block water molecules from entering the cell. So we have already discussed it in a previous question. The prokaryotic cell wall is composed of lipids and peptidoglycan. This is why they are more resistant to osmotic shock than eukaryotic cells. Permeability, ये सब हम already discuss कर चुके. These are present in eukaryotic cells. So here our answer is option A. Now moving next. The bacterial genus where sterols are present in the cell membrane is Vibrio, Mycoplasma, Escherichia, Chlamydia. So, sterols are present in cell membrane of Mycoplasma. So our answer will be option B. Now moving next, the bacterium that infects other gram-negative bacteria is Proteus mirabilis, Haemophilus influenza, Badillo vibrio, Pseudomonas tudia. So, which one of these bacterium infects other gram-negative bacteria? It is option C, Badillo vibrio. So our answer is option C. Now moving next. Which phase is used for phage display technique? T7, M13, lambda phase or pi 6. So phase display technique is a topic of molecular biology or microbiology that is not part of our syllabus, but it is usually asked which phase is used for phase display technique. So this is M13. M13 is used for phage display technique. So our answer will be option B. Now moving next, which of the following produce citric acid? So which of these will produce citric acid? Aspergillus, Pseudomonas, Saccharomyces, Clostridium. So citric acid is secreted by Aspergillus niger. That is our option A. Aspergillus. It secretes citric acids, or we can say it produces citric acid. So our answer will be option A, Aspergillus niger. Now moving next, which of the following organism is nitrogen fixing and found in rice fields in symbiotic association with Azolla? So we need to find that organism which is used in nitrogen fixing in rice fields, and it is found in a symbiotic association with bacterium Azolla. So that was also discussed in a old video. So what will be the answer? So Azolla is a fungi and symbiotic, and the symbiotic association is between Azolla and the blue-green algae Anabina. So they maintain a symbiotic relationship where जो algae part है हमारा, it provides nitrogen, and the जो हमारा फर्न वाला पार्ट है, एंड फंजाई जो है इट बेसिकली प्रोवाइड्स अ हैबिटेट फॉर द एल्गी. सो जो हमारी ब्लू ग्रीन एल्गी एनाबीना है ये उसके लिए नाइट्रोजन प्रोवाइड कराएगी अजोला के लिए, एंड अजोला जो है वो एनाबीना के लिए हैबिटेट प्रोवाइड कराएगा. एंड दिस प्रॉपर्टी ऑफ नाइट्रोजन फिक्सेशन has made Azolla a very important and economically important in the cultivation of rice. So our answer is option D. Anabina. It is a blue-green algae. Now moving next. Which of the following is used as a biofertilizer for soybean crop? So जो हमारी सोयाबीन क्रॉप होती है वहाँ पे बायो फर्टिलाइजर की तरह इनमें से किसे यूज़ करते हैं? राइजोबियम, एजोटोबैक्टर, नोस्टोक, एस्पेरिलियम. सो राइजोबियम जो होता है दैट इज यूज्ड एज बायो फर्टिलाइजर फॉर सोयाबीन क्रॉप. सो आवर आंसर विल बी ऑप्शन ए. Now here if you are preparing for TNMRB Food Safety Officer exam, you can enroll for this complete course where you will get full study material that includes 6 ebooks and a question bank. Also the test series based on exam pattern with all Tamil Nadu ranking, YouTube class notes, previous year paper, all the quality content only at 999, that is 1000 rupees. So you can find the link from description box and you can also download that from Play Store. Now the next question is, a common biopesticide for preventing plants from butterfly caterpillar is Trichoderma, Baculovirus, Glomus, Bacillus thuringiensis. So which one of these is used as a common biopesticides? जो प्लांट्स हैं उनको प्रोटेक्ट करने के लिए बटरफ्लाई के जो कैटरपिलर्स हैं उनसे प्रोटेक्ट करने के लिए. So which one of these is used as a biopesticide? So our answer is Bacillus thuringiensis, also known as BT. It is used in cotton, mustard and brinjal. So we have Bacillus thuringiensis, which is used as a biopesticide for preventing plants from butterfly caterpillars, and it is used in cotton, mustard and brinjal. BT cotton, BT brinjal and BT mustard, we have read about it. And next question is, yeast is used for the production of tetracycline, butanol, ethanol, citric acid. सो वी नो जो यीस्ट का जो हम यूज़ करते हैं दैट इज यूज फॉर द प्रोडक्शन ऑफ सिट्रिक एसिड. सो आवर आंसर विल बी ऑप्शन डी. नाउ मूविंग नेक्स्ट. विच एल्गे कैन बी यूज्ड एज फूड फॉर द ह्यूमन बीइंग? क्लोरेला, पोलिसिफोनिया, यूलोथ्रिक्स, स्पाइरोगायरा. सो विच वन ऑफ दीज कैन बी यूज्ड एज फूड? So chlorella is the mostly cultivated eukaryotic algae because it is used as health food or food supplement, and it is also used in pharmaceutical and cosmetic industry because it contains proteins, carotenoids and some immunostimulators, so which makes it a health food. So our answer will be option A, chlorella. Now moving next, which of the following microorganisms is not used as a biofertilizer? So which one of these is not used as a biofertilizer? So we have already discussed rhizobium is used as a biofertilizer, nostoc is also used as a biofertilizer, and so is mycorrhiza. It is agrobacterium that is not used as a biofertilizer. Agrobacterium is a disease causing microorganism. So our answer will be option D. Agrobacterium is not used as biofertilizer. Now moving next, find the incorrectly matched pair. Serratia, drug addiction. Spirulina, single cell protein. Rhizobium, biofertilizer. Streptomyces, antibiotic. So Serratia जो है हमारा ऑप्शन ए, that is incorrectly matched. कुछ Serratia is a disease causing gram-negative bacteria that causes disease meningitis, जिसे हम मिर्गी भी बोलते हैं. So that is caused by Serratia. So it is incorrectly matched pair. जो हमारा स्पाइरुलिना होता है, it is single cell protein, it is true. Rhizobium biofertilizer, आज की वीडियो में ही काफ़ी बार डिस्कस हो चुका है ये. स्ट्रेप्टोमाइसिस जो है हमारी एज अ एंटीबायोटिक वर्क करती है. सो ऑल दिस डिफाइंस द मेन फीचर ऑफ माइक्रो ऑर्गेनिजम्स एक्सेप्ट द ऑप्शन ए. सो आवर आंसर विल बी ऑप्शन ए. नाउ मूविंग नेक्स्ट. ट्राइकोडर्मा hergianum is useful for bioremediation of contaminated soils, gene transfer in higher plants, reclamation of wastelands, biological control of soil-borne plant pathogens. So Trichoderma herginium is a free-living fungi that is commonly used in controlling the disease-causing plant pathogens. So our answer will be Trichoderma herginium is useful for biological control of soil-borne plant pathogens. That is option D. So it is a fungus. Trichoderma herginium है हमारा. So it is also used as a fungicide. So now moving next. Which of the following organisms is used in alcoholic fermentation? Pseudomonas, Aspergillus, Saccharomyces, Penicillium. So which of these organisms is used in alcoholic fermentation? So Saccharomyces is used in alcoholic fermentation. So our answer will be option C, Saccharomyces. Now moving next. The target microorganism in canning is. So जो हमारी कैनिंग प्रोसेस होती है उसमें जो टारगेट माइक्रो ऑर्गेनिज्म है वो कौन सा होता है? क्लोस्ट्रीडियम बोटुलिनम, स्ट्रेप्टोकोकस थर्मोफिलस, PA3679, लैक्टोबैसिलस बुल्गारिकस. So it is PA369. It is also known as surrogate of Clostridium botulinum. So the target microorganism in canning is PA369. It is used to validate the thermal processing strategy used during the canning process to extend the shelf life of product and protect it from deterioration. So our answer here is option C. Now moving next. The desirable change in food that makes it unfit for human consumption is referred as food decay, food spoilage, food loss, all of these. So जो हमारे फूड में जो चेंजेस होता है which makes it not suitable for the consumption, that is known as food spoilage. जब हमारा फूड खराब हो जाता है तो वैसे कि it's not right to eat it, it can lead to several diseases. So it is referred as food spoilage. Now next is food preservation involves increasing shelf life of food, ensuring safety for human consumption, both A and B. So food preservation involves increasing the shelf life of food and as well as ensuring safety for human consumption. So our answer will be option C, both A and B. So now moving to the next question. Residues of alpha toxin M1 is found in dried fruits and nuts, cereals and grains, honey, milk and dairy products. So alpha toxin M1 is found in milk and dairy products. So our answer will be option D. सो जो हमारा अल्फा टॉक्सिक B1 होता है दैट इज फाउंड इन ड्राई फ्रूट्स एंड नट्स. सो जो अल्फा टॉक्सिक B1 होगा दैट इज प्रेजेंट इन ड्राई फ्रूट्स एंड नट्स, एंड अल्फा टॉक्सिक M1 इज प्रेजेंट इन मिल्क एंड डेयरी प्रोडक्ट्स, and alpha toxin B1, B2, G1, G2, these are present in cereals and grains and honey. B1, B2, G1, G2, and M1 is present in milk and dairy products. G1 and G2, so don't get confused in it. Now next question is, pasteurization is a low temperature treatment, steaming treatment, high temperature treatment, low and high temperature treatment. So we have discussed it multiple times. Pasteurization is a high temperature treatment, high temperature for shorter time. So this is high temperature treatment. So our answer will be option C. Now moving next. Normal bacteria stops division at 10 degrees Celsius, at 5 degrees Celsius, at 0 degrees Celsius, at 20 degrees Celsius. So when does a normal bacteria stops division? So जो normal bacteria है it will stop division at 5 degrees Celsius. So our answer will be option B. Now moving next. Which of the following statements are true with chemical preservatives? Microbial or microstatic agents. Chemical preservatives are often hazardous to humans. Sodium benzoate is widely used preservative. All of these. So this is what we have already discussed. Sodium benzoate is widely used preservative. So, and we also know chemical preservatives are often hazardous to humans, and these are microbial or microstatic agents. So our answer will be all of these. Option D is the right answer. Now moving next. Common food poisoning microbes are. So जो food poisoning होती है, so we have a very large number of reported cases of food poisoning. So which microbes causes it? Clostridium and Salmonella, Clostridium and E. coli, E. coli and Salmonella, Clostridium and Streptococcus. So which of these microbes causes food poisoning? Food poisoning is caused by Clostridium and Salmonella. So our answer will be option A. Now next is, botulism is caused by Clostridium botulinum, all Clostridium species, Clostridium tetani, Clostridium subtilis. So botulism is usually caused by the home canned foods, मतलब जिनकी कैनिंग ठीक से नहीं हो पाती, हम जो घर में कैंड फूड बनाते हैं. सो बोटुलिज्म इज कॉज्ड बाय बेसिकली दो फूड मटेरियल्स. एंड जब हम कैनिंग करते हैं सो हम कौन से बैक्टीरियम को किल करते हैं? सो वी किल क्लोस्ट्रीडियम बोटुलिनम. सो जो हमारा होम कैंड फूड होता है उसमें प्रॉपर कैनिंग नहीं हो पाती तो क्लोस्ट्रीडियम बोटुलिनम रह जाता है, एंड दैट कॉजेस botulism. So our answer will be option A. Now last question for today. Botulism prevention involves proper heat sterilization before food canning, addition of chemical preservatives, proper low temperature treatment before food canning, or all of these. So we just discussed botulism is caused by Clostridium botulinum, and जब हम कैनिंग करते हैं तो हाउ डू वी किल इट? टू हीट स्टेरिलाइजेशन दैट kills the Clostridium botulinum and hence prevents the botulism. So if we will do proper heat sterilization before food canning, we won't require anything else to prevent botulism. So our answer will be option A. So that was all for today. This is the answer key. It comes in your YouTube class notes where you will get this PDF, so you can practice and you can refer to this answer key for your answers to check your preparation level and to know about your weaker areas. So if you haven't enrolled for these courses yet, you can enroll for this course now. You can find the link in description box. And apart from them, these are the courses and test series available at Agriculture and GK, IBPS AFO, UKSSSC AAO, HPPSC ADO, NABARD exam, JKSSP Horticulture Technician, OPSC AAO, OPSC AHO, OPSC Assistant Agriculture Engineer, UPPSC Assistant Engineer Agriculture, ICR JRF, ASRB NET, ICR NET, Fisheries exams, Forestry exams, FSSAI exam, Tripura FSO, TNMRB FSO, TNPSC AO, AAO, DSSSB SO Horticulture, FCI AGM, Cotton Corporation of India exam. So you can find all these courses at Agriculture and GCAP. You can download it from Play Store and you can also visit the website agriculturegk.in. And for the free resources, you can subscribe the YouTube channel of Agriculture and GK. You can also join the Telegram group and follow us on Instagram. So that was all for today. Thank you for watching this video.